going to talk a little bit about the physics of focusing and the physics of light, and then we'll talk a little more, covering three sections in this video. So wavelength and color. Light is an example of electromagnetic radiation, energy in waves, long radio waves to short X-rays, and visible light occupies a very small portion of that spectrum. It specifically is between the 400 and 700 nanometer range wavelengths, and the eyes have receptors that are able to detect wavelengths of that range. So if we look at the spectrum, we see way down here gamma rays, radio waves. It's a very narrow band that we can actually detect with our eyes. Light can be thought of as packets of energy. They are referred to as photons or quanta. They travel in a wave-like fashion at very high speeds. When visible light goes through a spectrum, it's broken into bands of color we see as the rainbow. Red wavelengths are the longest, they have the lowest energy. Violet are the shortest, they have the most energy. Color that the eye perceives is a reflection of that wavelength. So grass is green because it absorbs all colors except green. White reflects all colors, and black absorbs all colors. So here are our photoreceptors. We have blue cones, green cones, red cones, and the rods actually respond to the strongest to about 500 nanometers. Refraction. Refraction and lenses are important because refraction is the bending of light, and that's due to a change in the speed of light when it passes from one transparent medium to another. The path of light is at an oblique angle, so from air to liquid we get refraction. And our lenses, and our lenses of our eye can refract light because they're curved on both sides. Something that is convex is thicker in the center than at the edges, whereas concave is thicker at the edges than in the center. So if you look at a straw in a glass of water, it looks like the two pieces of the straw are disconnected because of refraction. Convex lenses bend light passing through it so that they converge at a focal point. The image formed at that focal point is going to be upside down and reversed from left to right. A concave lens disperses light, which would prevent it from being focused. So if we have a point source here and it goes through the convex lens, it would focus. This point would be focused here. This point would be focused here. So in reality, we get a real image on the back of our eye, on the retina of our eye, and that real image is upside down and reversed, and our brains work to correct that. Pathway of light is entering the eye, has to pass through the aqueous humor, the lens, the vitreous humor, the entire neural layer of the retina, and then it makes it to the photoreceptors. And there are three places along the way where light is refracted: when it goes into the cornea, entering the lens, and leaving the lens. Now, the majority of the refractory power is in the cornea, but it's constant, it can't change. So if we want to change in order to focus, we have to use the lens. The lens is able to adjust its curvature and to allow for fine focusing. It can focus for distant vision and for close vision. The ciliary muscles and the ciliary zonule focus an image by changing the shape of the lens. They're arranged like in a sphincter-like structure around the lens, and they contract, which would loosen the zonule fibers, and relaxation of them tightens the fibers. So our eyes are best adapted for distant vision. Our far point of vision is the distance beyond which no change in the lens shape is needed for focus, and that's typically 20 feet for an average eye, an emmetropic eye. cornea and the lens focus light precisely on the retina at that distance, which is why when we do an eye test, we say 20/20 is normal. The ciliary muscles are completely relaxed in distant vision, causes pull on the ciliary zonule. As a result, the lenses are stretched flat. So when we look at something 20 feet away, our muscles are relaxed. Image gets refracted here, here, and here, and we end up with what is known as a real image on the retina, and that is upside down and backwards, and then the brain translates it into what we are looking at. For close vision, less than 6 meters away, the objects are going to diverge as they approach the eye. So we're required to make active adjustments using 3 simultaneous processes. Accommodation of the lens, which changes the lens shape to increase refraction. So for near point vision, the closest point would be defined as the closest point on which the eye can focus. Presbyopia is a loss of the ability to do that accommodation. This says over the age of 50, it gets maybe quite a bit younger than that before you start realizing that you have to hold things further away in order to see them clearly. We also constrict the pupils, so accommodation of the pupillary reflex involves constricting the pupils. That prevents the most divergent light rays from getting into the eye. That's mediated by the parasympathetic nervous system, and also convergence of the eyeballs. So our eyes, rather than moving together, focus towards an object. So the left one looks to the right one looks to the left. And that's controlled by the somatic motor neurons innervation in the medial rectus muscles. So the eye is going to bulge for close vision. That has to do with the parasympathetic input to the ciliary muscle, loosening the ciliary zonule, allowing the lens to bulge. So that leads to nearsighted, farsighted. Myopia is nearsightedness. The eyeball is just a little too long, so the focal point ends up being in front of the retina. That can be corrected with a concave lens. Hyperopia, or farsightedness, the eyeball is a little short, so the focal point would actually be behind the retina, and that can be corrected with a convex lens. Another issue with lenses potentially is called astigmatism, in which there's an unequal curvature in different parts of the lens, and that can be corrected with cylindrically ground lenses or laser procedures. So emmetropic eye, 20 feet away, focus perfectly on the retina. In a myopic eye, the lens is a little longer than it should be, so the focal point would be here, the retina is back here, and you use a concave lens to move that focal point to the lens, to the retina, excuse me. So in a hyperopic eye, the eye is a little short, and so the focal point actually would be behind the retina, so the image is still converging, going through the retina. That would be corrected with a convex lens. All right, so how does that light lead to nerve transmission? That is phototransduction. So photoreceptors, the rods and cones, are modified neurons. They resemble upside-down epithelial cells. They have a cell body, a synaptic terminal, and 2 segments. The outer segment is the light receiving region. It contains the visual pigments, which we'll call photopigments, and those pigments are going to change shape as they absorb light. The inner segment joins to the cell body. The inner segment is connected via cilium to the outer segment and to the cell body through the outer fiber. Cell body is connected to the synaptic terminal through the inner fibers, and the plasma membrane of the outer segment folds back to form many discs. Those photopigments are embedded within the discs. Photoreceptors are vulnerable to damage. They degenerate if the retina becomes detached. They are destroyed by intense light. So vision is maintained because the outer segment is renewed every 24 hours. The tips fragments fall off and get phagocytized by other cells. So here's a photoreceptor. This is our cell body. The inner fiber and outer fiber is the inner segment, and the outer segment where our discs containing photopigments are found. So rods are very sensitive to light, making them best suited for night vision and peripheral vision. They contain a single pigment. There's only one kind of pigment, so the vision is perceived in gray tones only. Their pathways also converge, which means that we get fuzzy, indistinct images. There's many as 100 rods can converge into one ganglion, so we don't get nice sharp edges when using our rods. The cones are less sensitive to light, so they require bright light in order to be activated. They react more quickly than rods. They have one of three pigments that allows us to see colored or have colored vision. They also have non-converging pathways, so we get detailed high-resolution vision. Some cones might have their own ganglion cell, so we can put them together and we get very sharp edges this way but in the process of interpreting that image. So you should go over figure or table 15.1 for a comparison of rods and cones. Color blindness is the lack of one or more of the cone pigments, generally inherited as an X-linked condition, so it is more common in males. Approximately 8 to 10% of males have some form of color blindness. The most common type of color blindness is red-green, in which either the red cones or the green cones are absent, and depending on which cone is missing, red can appear green or green can appear red, and individuals rely on different shades to get cues of the color. The pigments include retinol, which is a light-absorbing molecule that combines with one of four proteins. Those proteins are called opsins, and so the combination of retinol and opsins forms a visual pigment. They are synthesized from vitamin A. The four opsins are rhodopsin, which is found only in rods, and in cones we have green, blue, or red opsins. So the cone wavelengths overlap, so the same wavelength can trigger more than one cone, and that enables us to see the hues of color. So yellow light is going to stimulate red and green cones, but if more reds were triggered, then it would appear orange to us. So orange would trigger more red than green. There are isomers of these retinols. Isomers, if you remember from chemistry, are differences in three-dimensional form. Cis or trans is a common way to describe them. So when retinol is bent in the dark, but when it absorbs light, it straightens out. The bent form is called cis, so it's 11-cis retinol. Whereas the straight form is called all-trans retinol. So conversion of bent to straight is going to initiate the reaction that leads to the electrical impulses along the optic nerve. So phototransduction is the process by which the pigment captures a photon of light energy and that is converted into a graded receptor potential. So capturing the light, the dark, deep purple pigment of rods is rhodopsin. They're arranged in the rod's outer segment. There are 3 steps in rhodopsin formation and breakdown: pigment synthesis, pigment bleaching, and pigment regeneration. Similar process in cones, but different types of opsins and cones require more intense light for it to happen. So we'll focus on rhodopsin. So the pigment synthesis, opsin and 11-cis retinol are going to combine to form rhodopsin, and that's the rhodopsin we have in the dark, so 11-cis. When rhodopsin absorbs light, the 11-cis isomer of retinol changes to the all-trans isomer of retinol. So that would be the beginning of photo-bleaching. When 11-cis becomes all-trans, the retinol and the opsin separate from one another. So the rhodopsin breaks down. To regenerate that, the all-trans retinol has to be converted back to 11-cis retinol, and then it will recombine with the opsin, regenerating rhodopsin. So in the dark, 11-cis, when light hits it, a cis becomes trans, the retinol and the opsin separate, and then to regenerate it, we convert it back to 11-cis and combine it with opsin. So we go from rhodopsin to opsin and retinol. Rhodopsin in the dark. As light comes in, the opsin and the trans retinol separate from one another, and that is a cycle that goes on. So light activated rhodopsin then is going to work on second messengers that activate a G protein. This one is called transducin. Transducin is going to activate an enzyme PDE that's going to break down the cyclic GMP. In the dark, cyclic GMP holds cation channels of the outer segment open. Sodium and calcium enter and depolarize the cell. When the light causes cyclic GMP to break down, those channels close, the cell hyperpolarize, and it's hyperpolarization that's going to be the signal for vision. So the cell is depolarized at rest and hyperpolarized for vision. So again, retinol absorbs light, that changes its shape, separating the retinol from the opsin. That retinol causes a G protein called transducin to become active. Transducin binds to PDE, phosphodiesterase, causing phosphodiesterase to take cyclic GMP and make it into the linear form of GMP. And the lack of cyclic GMP is going to close these channels, and that closing those channels causes the cell to hyperpolarize, and the hyperpolarized cell begins the signal transduction process. Photoreceptors in bipolar cells only generate graded potentials, not action potentials. When light hyperpolarizes photoreceptor cells, they stop releasing the inhibitory neurotransmitter glutamate to bipolar cells. Bipolar cells, which are no longer inhibited, can then depolarize. They release neurotransmitter onto the ganglion cells. It's the ganglion cells that generate the action potentials that get transmitted to the optic nerve of the brain. So cyclic GMP channels, gated channels open, that allows calcium influx, and the photoreceptor depolarizes. The voltage-gated calcium channels open in the synaptic terminal. The neurotransmitter is released continuously. That neurotransmitter causes graded potentials in bipolar cells, resulting in hyperpolarization. And the hyperpolarization closes the calcium voltage-gated calcium channels, which inhibits the release of neurotransmitter, and the neurotransmitter inhibition causes excitement in the ganglion cell. That's where we would get light. So here the gated channels are closed. Photoreceptor hyperpolarizes. The hyperpolarized does not any longer release neurotransmitter onto the ganglion cells. The ganglion cells would then release neurotransmitter, and an excitatory potential would happen in the ganglion cells. We can very quickly adapt to light and dark conditions. So rhodopsin is sensitive that bleaching occurs even in very dim light. In bright light, the bleaching occurs so fast that rods are virtually non-functional. Cones respond to bright light. So activation of rods and cones depends on light adaptation or dark adaptation. Light adaptation happens when moving from darkness into bright light, and that leads to a glare because for a moment or two, both the rods and cones are strongly stimulated. A large amount of pigments are being broken down instantaneously, producing the glare, and the pupils will constrict. Over the course of about 5 to 10 minutes, the rods will turn off, the retinol sensitivity will decrease, and the cones and neurons will rapidly adapt. The other thing, dark adaptation, happens when moving from bright light into darkness. It seems very, very dark for a while because the cones stop functioning in the low light intensity. The bright light had already bleached the rod pigments, so they're still turned off. That will cause the pupils to dilate. But rhodopsin will begin to accumulate in the dark, so the retinal sensitivity starts to increase, and transducin returns to the outer segment, and this sensitivity increases within 20 to 30 minutes. Night blindness or nyctalopia is a condition in which rod function is hampered. That can lead to the ability to drive safely at night being impaired. It could be due to rod degeneration, which is commonly caused by a prolonged vitamin A deficiency. If you get that early enough, vitamin A supplements can help restore that function. It can also be triggered by a disease called retinitis pigmentosa that we mentioned last week in lecture. The degenerative retinal disease that destroys rods. And when you have this, the tips of the rods are not replaced and they slough off. All right, last section is what do we do with the signals? How do we process them, and how do they get to become visual information? So far we've talked about light being bent, then hitting receptors, and those receptors depolarizing to electrical signals. So how do those electrical signals become our vision? The axons of the retinal ganglia form the optic nerve. The medial fibers from each eye crosses over at the optic chiasma and then continue on as optic tracts, which means each optic tract contains fibers from the lateral aspect of eye on the same side and the medial aspect of the opposite eye, and each carries information from the same half of the visual field. Most of the fibers of the optic tract continue on to the lateral geniculate nuclei of the thalamus. From there, the thalamic neurons form optic radiation, which projects to the primary visual cortex in the occipital lobes, and this is where we get conscious perception of the visual images. There are other optic tract fibers that send branches to the midbrain. One set ends up in the superior colliculi, an area that's controlling extrinsic eye muscles. A small subset of the ganglionic fibers in the retina contain melanopsin, a circadian pigment, which projects to the pretectal nuclei, which is involved in pupillary reflexes, and the suprachiasmatic nucleus of the hypothalamus, which is a timer for our daily biorhythms. So here those pathways are mapped out. And again here with a brain to compare them to. Depth perception results because both eyes see the same image from slightly different angles, and the visual cortex fuses those together with they're slightly different images, so we get a three-dimensional image that leads to our depth perception if we have two very good working eyes. Loss of an eye or destruction of one optic nerve will eliminate true depth perception entirely. The peripheral vision on the damaged side is also affected. If neural destruction occurs beyond the optic chiasma, then part or all of the opposite half of the visual field is lost. For example, strokes can affect the left visual cortex, which leads to blindness in the right half of the visual field. Retinal cells split input into channels that include information about color, brightness, angles, direction, speed, movement, edges. Lateral inhibition decodes the edge information. That's the job of amacrine and horizontal cells. Ganglions pass the information to the lateral geniculate nuclei of the thalamus to be processed for depth perception with cone input emphasized. The primary visual cortex contains a topographical map of the retina. So neurons respond to dark and bright edges and orient the object. They provide form, color, motion inputs to the visual association areas. Info is also passed on to the temporal, parietal, and frontal lobes, where objects are identified and location in space is determined. And that will cover sections 2, 3, and 4.